So before we look at aerobic respiration, which requires oxygen and occurs in the mitochondria, let's first look at the structure of a mitochondria. So the mitochondria has two membranes. You can see it, the brown outer membrane. Let's go with the outer membrane. And then the folded stuff on the inside is the inner membrane of the mitochondria. So when we look at the mitochondria structure, we can see here there's an outer membrane and an inner membrane. Now inside of that inner membrane, we call that area the matrix. So this space, that's a bluish green color, is the intermembrane space. Here's our inner membrane and our outer membrane. Now this is important to note that right here, the inner membrane is where the electron transport chain is located. Yep, it's located there. All right, so then in the matrix is where the citric acid or the Krebs cycle occurs. And because the inner membrane is where the electron transport chain is, we know from photosynthesis that electron transport chains pump hydrogen. And so the hydrogens that are going to be pumped in the E2C will accumulate in that intermembrane space. So if you're doing your boxes at home, you can draw some box 5. Okay. So when we look at what follows glycolysis, if oxygen is present. So the key here is glycolysis occurred in the cytoplasm, and if there's oxygen, we will use the mitochondria to further oxidize our food and gain the energy available in the bonds of our pyruvate. So the next step, we'll use that ATP. The next step, we're going to zoom in here to the mitochondria, and we're going to look at the oxidation of pyruvate. Now think, oxidation means to break the bonds and take the electrons. So pyruvate is going to be giving electrons. So when we oxidize a molecule, something is going to be reduced. So here, in this 3-carbon pyruvate that has potential energy still stored in those bonds, we're going to break it to take some of the electrons. We're going to break a bond and take the electrons. That electron carrier is going to be reduced. Well, it just so happened we broke the bond and we took the electrons in the bond that was connecting that 3rd carbon. So now that 3rd carbon is going to come off as carbon dioxide. So again, let me play that one more time for you. Pyruvate has 3 carbons. We're going to oxidize it, which means to break the bonds and take the electrons. So we're going to break the bonds, take the electrons, and reduce an electron carrier with them. Oops, that was fast. We're going to take the electrons and reduce an electron carrier, NADH. Now, carbon dioxide is released. One of the 3 carbons is broken off as carbon dioxide, and it'll actually diffuse out of your cell. Wait, there it is. And then there's a helper enzyme. We call this coenzyme A. And coenzyme A is going to help acetyl enter into the Krebs cycle. So this molecule called right here, oh, can't really see that yellow, but this molecule here is called acetyl-CoA. So acetyl-CoA still has some remaining potential energy in these 2 carbons, and so in the next step called the Krebs cycle or citric acid cycle, we will further oxidate the remaining potential energy. Oh, sorry. We will oxidate that food that's remaining, gaining the energy in the form of electron carriers. Okay, so this is going to actually happen twice because there's 2 pyruvates. All right, so let's go ahead and see what happens next. So now we're moving on to the citric acid cycle or the Krebs cycle, and then eventually the electron transport chain. So right now we can kind of summarize. In glycolysis, we got 2 pyruvates, but we also got 2 NAD. We got 2 NADHs, 2 electron carriers, right here, and a DH. And then we got some electron carriers, reduced electron carriers made in the oxidation of pyruvate. And now in the citric acid cycle, we will continue to make reduced electron carriers, and all of those electron carriers are going to carry the electrons over to the electron transport chain. Okay, so here we go. When we look at the Krebs cycle, here we have the products of what we've seen so far. 2 of these NADHs were made from glycolysis, and then this was from the oxidation of one pyruvate, the oxidation of another pyruvate. So here we have this coenzyme A that is like a helper protein to help acetyl enter into the Krebs cycle and start the process of completely oxidizing our food. So as we look to see, oh, we talked about that. Good job, good job. Okay, so here we go. The first step, we're going to take this 2-carbon acetyl and we're going to attach it to a 4-carbon molecule. This 4-carbon molecule is called oxaloacetate. Oxaloacetate, doesn't really matter. And so at the end of the Krebs cycle, we're going to need this oxaloacetate all over again. So the next acetyl-CoA can come in. So this Krebs cycle is a cycle. It's going to move through a series of steps where at the end of it, we will have oxaloacetate again. So that way it can continue. So when we look at this, the first step, though, is going to be taking acetyl, and an enzyme is going to attach it to oxaloacetate, which is 4 carbons. So here we have our 4-carbon oxaloacetate and our 2-carbon acetyl. We're going to form a 6-carbon citrate. So here we have our citrate. This is also why it's called the citric acid cycle. And now when we look at like the point of this, remember, the whole big picture is to fully oxidize our food. So we had 2 carbons left in our acetyl. So we need to oxidize that fully. So here we go. So our first step, there'll be an enzyme that's going to oxidize this 6-carbon molecule, going to break the bonds, take the electrons, so that's going to be oxidized. The electrons will be reduced onto an electron, or put onto an electron carrier. So that electron carrier is reduced. So you see an oxidation-reduction reaction. We do it again one more time. So here now we have 5 carbons. And these 5-carbon molecule, we're going to take one of the, carbon dioxide, oxidation-reduction. All right. So now we see this carbon and this carbon. We also have the carbon dioxide from the oxidation of pyruvate. Here we have 3 carbons. Those 3 carbons were the 3 carbons that we found in pyruvate. If you have 2 pyruvates, that means you're going to get 6 carbon dioxides. So if you think about photosynthesis, they required 6 carbon dioxides to build a glucose. And here we've broken it down, our glucose, back into the 6 carbon dioxides the plants use to build. So by the end of the Krebs cycle, we will have fully oxidized our food. Our food molecule, our glucose, is now completely turned back into carbon dioxide. And all of the bonds that the plant built during photosynthesis have now been broken or oxidized. We've oxidized our food, taken the electrons, and we've reduced electron carriers. So all of that energy that was stored in our foods, not all of it, some of it's given off as heat, but a lot of the energy that was in our food is now in the form of electrons on electron carriers. The electron carriers are going to carry that energy to the electron transport chain where we can convert it into ATP, which is usable energy for us. All right, so, but there's a problem. So while we have oxidized our food, our carbons from our glucose molecule are now all in the form of carbon dioxide. We have the problem that this right here is not oxaloacetate, the 4-carbon molecule that we started with in the Krebs cycle. So there's actually about 5 more steps involved in the Krebs cycle or citric acid cycle. So along those next steps, it's basically turning this 4-carbon molecule here back into oxaloacetate. So along the way, we'll actually make 1 ATP by substrate level phosphorylation. And in relation to all of life, 1 ATP is pretty kind of insignificant. The majority of our ATP will be made in the electron transport chain, but we still make 1 by substrate level phosphorylation. And then there's other enzyme-controlled reactions that are going to be happening here in the Krebs cycle. And so this we haven't seen before. This is a new electron carrier. It works the same as NAD+, or the NADP+ we saw in plants in photosynthesis. So it's still an electron carrier, just a new one. And in one of the steps of turning these 4 carbons back into oxaloacetate, this electron carrier will actually be reduced and gain some electrons and hydrogens. And then there's one more electron carrier, NAD+, that will be reduced in the process of converting that 4-carbon molecule back to oxaloacetate. So here we have our 4-carbon oxaloacetate that we can then begin the Krebs cycle all over again. So then you would have the new acetyl-CoA come in, attach with the enzyme, forming citrate, and then we can oxidize this. So when we look at the big picture, we started with glucose with 6 carbons, then we had pyruvates, then we oxidized pyruvates, and we had acetyls with 2 carbons left, and then we oxidized those, and then our our carbons are all now in the form of carbon dioxide. But here, the oxidation of pyruvate and the Krebs cycle, they both happened twice because there was those 2 pyruvates. So now, this acetyl right here is the second pyruvate, or because of the second pyruvate. So here it's our second acetyl-CoA, so we'll go through the Krebs cycle again. And so, as we go through the Krebs cycle again, we make even more electron carriers, and at this point, our food has been fully oxidized, broken up into carbon dioxide gas, and all of the energy that we could possibly get is put onto a reduced electron carriers in the form of electrons. Granted, some of our energy, quite a bit of our energy, when we broke those bonds were released as heat, which is not a usable form of energy for us, but enzymes have evolved to control these reactions to try to limit how much of that potential energy is lost as heat. And that is the end of our Krebs cycle. Okay, good job.